Thank you all for standing by and welcome to our webinar entitled Finding Veterinary Pharmaceuticals in Lake Erie Tributaries. This webinar series called Freshwater Science will highlight Ohio's Sea Grant and partnering scientists every month. Every quarter is a different focus, from human health and fish farming to harmful algal blooms and human decision-making, bringing applied research to the public on issues that affect our Lake Erie communities. I am Jill Gentis-Banicki from Ohio Sea Grant and Stone Laboratory, and joining me today is Dr. Laura Johnson from Heidelberg University. Laura Johnson is the director of the National Center for Water Quality Research at Heidelberg University in Tiffin, Ohio. At the center, long-term monitoring of streams and rivers is used to examine the influence of human activities on water quality and help decide actions that lead to healthier ecosystems. Dr. Johnson is best known for research examining the linkages between agricultural runoff and harmful algal blooms in Lake Erie. As director, she has overseen the expansion of the monitoring program, curriculum development in watershed science, revitalization of the Sandusky River Watershed Coalition, and has been part of numerous work groups and advisory boards. We're delighted to have Dr. Johnson here today to talk to us about her research. Before we do that, though, I'd like to talk a little bit about our webinar in general. During our presentation, all participants will be in a listen-only mode. Afterwards, around 12:20, I will conduct a question and answer session. If you'd like to ask a question during the presentation, please feel free to pull up that chat feature anytime during her talk, and I will collect and pose your questions out to Dr. Johnson at the end of her presentation. As a reminder, this webinar has auto captioning and is being recorded to be posted later onto our website. Also, we will post a webinar survey in the chat feature toward the end of the half an hour. Please take a few minutes after the webinar to fill out that survey. It always helps us continue to bring you great webinars. So without any further delay, I'd like to introduce Dr. Johnson, presenting Finding Veterinary Pharmaceuticals in Lake Erie Tributaries.
Awesome. Thank you, Jill.
And let me see, all right, you got it.
All right. You see everything you need there?
Yes.
And you're in presenter mode.
There you go.
Perfect. Thank you. All right. So hi, everyone. As Jill said, normally at Heidelberg, I am presenting on the water quality in terms of nutrients and sediments, making it into our rivers and streams, especially the ones that go into Lake Erie. But this one's a little different. So for this talk, I'll be talking a bit more about veterinary antibiotics that we've been finding with a more specialized study, looking at these compounds in a lot of the stations where we conduct monitoring. I do want to note that I couldn't have done any of this work without our help of Jake Beeler and Nate Manning here at Heidelberg, as well as Dan Snow, who's in Nebraska, that did and managed all of the sample analysis. So let's jump right in. Why should we be thinking about this at all? Well, the first reason, especially thinking about veterinary antibiotics or antibiotics that are used on livestock or in animal agriculture, is because antibiotics are water soluble, so they run off easily. They're also excreted in urine or feces, usually without really any changes. And then oftentimes manure is then applied to land. So there's a pathway in which antibiotics can enter our environment fairly easily. And we worry about that primarily because of the development of antibiotic resistant bacteria. We know that, using antibiotics on food animals can enhance these resistant strains that are present in human medicine. We already know that there's some antibiotic resistance in foodborne pathogens like Campylobacter, Salmonella, and E. coli that make it harder if we end up getting E. coli from food to be able to treat because they're resistant to a lot of antibiotics. And then we also know that this resistance is easily transferred among different bacteria, and that that antibiotic resistance can enter our environmental bacteria and sort of change how our ecosystems are functioning as well. Antibiotics are also a human health risk, and on their own, a lot of times people are allergic to antibiotics, and so having them around in the environment poses a risk for those folks. And also they can be harmful to aquatic organisms. Some of them at somewhat low levels can cause harm to especially some plants and animals. So why are antibiotics used in livestock in the first place? Well, I think that's pretty obvious. If you have, for example, from this picture here, a cow that has an infection, then you're going to need to treat it with antibiotics because that is the most humane and ethical thing to do for that cow. So it's usually to treat illnesses. Sometimes you can see where we have an illness that's spreading throughout a herd or a flock, that antibiotics are used a little bit more widespread to prevent illnesses going into other, other animals that are sort of being infected at that same time. A lot of times you also can see that antibiotics are used as a growth promoter. It turns out if you add those to feed, it improves the efficiency at which that feed has been able to take it up, and that then livestock are able to grow faster and get larger. So that's the other mechanism that we see antibiotics used. However, it's important to note antibiotic use in livestock is being increasingly regulated. Since 2017, using antibiotics that are commonly used in human medicine are not allowed to be used for growth promotion for any animals that are associated with food production. And that's to sort of basically help protect sort of our human medicine on one side. And many antibiotics can only be used under the guidance or under prescription, essentially, of a veterinarian. And then starting this year, any remaining over-the-counter antibiotics will become prescription only. I put in here except for ionophores. So it's important to note that while we're seeing increasing regulation, there's always some loophole or caveat of some form of an antibiotic that maybe isn't being used for human medicine or something like that, that is now taking some of these other antibiotics' place. And so there's still usage, and there's still some questions about that. Now, when we are in the Western Lake Erie Basin, we know that there is quite a bit of animal agriculture all throughout the basin. This is a map from the Environmental Working Group. They did a lot of work sort of pulling together information on the locations of different types of animal operations. The orange ones being the larger facilities that have enough animal numbers so that they require a permit. The purple ones being the smaller facilities that don't have quite that many animals, they don't need the permit. And you can see there's some variation in the locations and the density of operations throughout the Western Lake Erie Basin, but it's fairly widespread. The other thing that we know is that we've been seeing increases in the number of animals, especially in the Maumee River watershed. That's what this example is. And this is data on the animal units of cattle and hogs in the Maumee River from the Ag Census. And since around 2000, we've seen sort of a steady increase in the number of animals in the region. So we have widespread livestock and increasing intensity of animals in this area. So that leads to the big question is, which one is sort of leading to antibiotic, potential antibiotic runoff, or maybe reductions in antibiotic runoff. We know we have these increases in livestock operations, but we also have new regulations. So are we still seeing veterinary antibiotics in our tributaries? How prevalent are they? Is it something we should be concerned about? And that's sort of what we want to touch on today. To do this, we're going to be looking at three main things here. So we're quantifying just the presence of antibiotics, hopefully their sources, as well as their concentrations in Lake Erie tributaries, trying to determine if we can look at those to get an understanding of how impactful, what the burden of animal agriculture is on different watersheds. And our final sort of fleeting hope here is that we might be able to then use that information to understand why we see variation in nutrient loadings from these different watersheds to Lake Erie. So if we see higher phosphorus in this one watershed, is it because there's a lot of animals, or is it something else? So to do this work, we sampled 3 locations for 3 years here. So 2020, 2021, and 2022. I will note 2020 was the COVID year, so we got a slow start. We only got a couple of samplings in that year. And we started here in the Sandusky River watershed. So you can see we monitored the Sandusky, Honey Creek, and Rock Creek here. And these were good choices because there's some variability. Sandusky and Honey Creek both had similar amounts of animals, slightly differing amounts of hogs versus cattle. But when we go to Rock Creek, you can see there was a lower total number of animal units, but a slightly higher amount of cattle. So we had some variation in animal units that we were across these 3 watersheds. But then in 2022, we decided to do a more broader survey across different watersheds that we monitor. And so you can see we added in a lot of different sites here, and that increased the variability in animals in these different watersheds. One of the sites I'll probably talk about a bit is Wolf Creek, which is up here in the Toledo area. And in Wolf Creek, that's primarily residential, so there are no animals, all the way up to, the Maumee that had, Sandusky and Honey Creek that all had fairly high animal units per kilometer square. So what do we do at these sites? Well, we deployed a passive integrated sampler. They're called POSIS. And basically, it's this little membrane. You can see the membrane in the cage here. You can see it deployed in this picture here. We would deploy those for about 21 to 28 days in each stream. And then we go and retrieve them, then redeploy them usually at that time through the growing season. And what this does is this slowly samples those antibiotics over an integrated period of time. So this is nice because we get a longer viewpoint of the potential amount of antibiotics in the water. And then when we would go to retrieve those samples, we also did a grab sample. So we had that to compare to this more integrated view. Samples were then sent to the University of Nebraska and Lincoln's Water Science Laboratory, and they were analyzed using LC-MS. For the POSIS samples, the sampler, they would have to be extracted before analysis with LC-MS, and then the grab samples required a slightly different process, but nonetheless, they were all analyzed there, and they did a fabulous job. And we analyzed for all kinds of various classes of antibiotics. I put the whole list here because I figured maybe some of you would want to see them all. But you can see there's a lot to choose from in terms of doing this analysis. And there was an awful lot of tongue twister names in here. So I apologize in advance if I butcher any of those. So let's just jump right in. What did we find? So these are our grab samples. And basically what I'm showing you here are all of those different compounds that I just mentioned. I'm going to turn this into a pointer so you guys can see my pointer instead. So all of the different compounds here, and this is the frequency that we were detecting them in our grab samples. So you can see I highlighted the ones that had the highest frequency of detection. That would be erythromycin, lincomycin, sulfamethoxazole, and thiobendazole. And of these, really, they were all veterinary compounds except for sulfamethoxazole, which is a known human compound, although in some digging, I think it can be used sometimes in animals as well. It's just not as prevalent. There were some compounds that we never detected in our grab samples. So that's important to note. You can see a lot of zeros or very low percentages here, and some that were somewhere in between as well, like monensin or sulfamethazine. Our highest concentrations found were for sulfanilamide and sulfamethoxazole, which aren't shown here in terms of concentration. But most of the time, all of these concentrations are far lower than those. So how do they compare to others? This is, I pulled data from a global synthesis study from 2013 that was looking at a lot of the similar compounds, and I listed those here. So these aren't all of the same ones, but they're a list of similar ones. And they included the median concentrations and their detection frequency. So I included our maximum concentration and detection frequency as a comparison. Now, it's important to note that this is global. So this includes places outside of the US where sometimes concentrations can be really high, like India and China. And so we do see that in our study in general, I put the ones in blue that were lower and only the ones in red that were higher. But in general, all of our concentrations tended to be lower than what was found in this global synthesis. That could also be because of the difference between 2013 and 2022 when we did the study, right? There's a little bit of difference in years there. But we did have a couple that were a little bit higher, sulfadimethoxine and sulfamethoxazole here. And then you can see there are a couple where we had more detection frequency. So these were like erythromycin and lincomycin, where we detected 84% of the time, where there was a little bit lower in this global study. To me, this indicates that, things don't look awfully different. We're probably a little lower, although because we're purposefully studying areas with lots of, that is animal agricultural focused, we probably saw higher detection frequencies for those animal antibiotics for a reason. So let's move into our passive samplers. So the POSIS samples, what do we find there? Somewhat similar, but slightly different results here. Our most consistently detected compounds here ended up being lincomycin, which was detected 98% of the time. Thiobendazole we detected 100% of the time. And then sulfamethazine, which is also another veterinary antibiotic, was detected 82% of the time. You see we had a couple of human antibiotics that were detected at high frequency of time as well. There is still, again, a number of things that we never detected or were at very low levels of detection frequency, which is, a good sign, but these very high detection frequencies is a bit concerning. The highest concentration we found was actually for penicillin G, which had a pretty low detection frequency, only 12%. And that was like our one Honey Creek site ended up one time having a lot of penicillin G in it for some reason. That is both used for veterinary as well as humans, but most commonly, I think it's more commonly found in animals. And then our second highest was sulfanilamide. So moving forward, I want to show you some figures over time with some actually our sites and not just the percent detections, because that gets a little tedious. So what I'm going to do is I'm focusing on our most consistent veterinary specific antibiotics that we found. I have them listed here. So we have lincomycin, monensin, sulfamethazine, and thiobendazole that I'm going to show you our series of figures here. So the first one I want to talk about is how do these antibiotics vary over time from Honey Creek, Rock Creek, and the Sandusky River watersheds. And just to orient you here, so this is showing our POSIS concentrations over time. I have Honey Creek in blue, Rock Creek is in red, and the Sandusky River is in purple, because it's the two of them combined. And what you can see here is a lot of variation. There seems to be maybe a slightly upward trend through the 2021 growing season in terms of the detection of monensin, but in general, it's pretty variable. It wasn't like one creek was always higher than the other, or that the Sandusky was always higher than the creeks. There is just, there's always something there, except for maybe two of these periods of, these 2 points here, and highly variable. We see the same thing when we look at the remainder of our different compounds as well. So lincomycin, you can see we had slightly higher detections here in December of 2020, similar with sulfamethazine, so potentially following some fall application of manure. But in general, these were all fairly variable. And basically flip-flopping on terms of what watershed had higher concentrations throughout the year. I do want to note that this lincomycin was a very high concentration and sulfamethazine is very low concentrations respectively. So if I put all of these on the same scale, you can see now that these look a little bit more similarly variable, monensin and lincomycin, and you can see that sulfamethazine was actually a fairly low concentration, even though it was detected fairly frequently throughout the time period. So question 2, how did the antibiotics vary across watersheds in 2022? So now we're looking at spatial variation instead of temporal variation. And remember, when we look across our watersheds, this is where we had differing amounts of animal units per square kilometer. So imagine this, and I'm going to show you now some figures of the same format here. So essentially, they were pretty variable, The monensin numbers here, Tiffin River had by far the highest amount. It was actually up at 25. I have it sort of cut off here so we can compare across these sites a little better. They didn't necessarily always match the number of animals that we had per watershed. You can see Tiffin River is a little higher here for lincomycin as well. There's some variation across the different sites. Sulfamethazine was lower than these other compounds, similar to what we found when we compared in Rock, Honey, and Sandusky over time. So that wasn't a huge surprise there. I do want to point out, though, that Wolf Creek had detectable amounts of veterinary antibiotics in a few of these cases, which is interesting since it's mostly residential. So the question is, well, maybe there's something coming in from somewhere else, or perhaps maybe there's some like small animal antibiotics that are being shared amongst these as well. So how do this is great and all, but this doesn't tell us about how this relates to the number of animals in the watershed that you would see here. So how do we better do that? Well, we always go into doing something like a regression approach. So let's look at how these two things scale together. And so if we look at now these same 4 compounds, but scaled to animal units, you can see that generally by having livestock in numbers, it helps describe some of that variation. It's not perfect, but it definitely helps. So for Monenson, we found that there's certainly increases, especially this one high point tended to have the higher total amount of cattle and poultry added together in terms of animal units. For lincomycin, it was all three, hogs, cattle, and poultry. You see a pretty good relationship here where we had higher lincomycin, where we had these higher number of animals. Thiabendazole was not very well related because there's this one really high point here where we had fairly low amount of cattle. But if we threw that out, then there was a little bit better relationship. At a minimum, I think you could say once you get over 5 animal units per square kilometer in a watershed, we tend to see higher amounts of thiabendazole. For sulfamethazine, there was a relationship with hogs, cattle, and poultry again. So from this, it makes me wish I had maybe a lot more watersheds to compare against, of course, or at least the longer term data to be able to do these comparisons, but this is somewhat promising. This is what we were hoping that we would find would be some relationship between compounds and what's in the watershed. I did want to point out one compound that I haven't been focusing on, and this is thiamulin. I have no idea how you say that, but I'm going with it. And you can see that this is a compound that's usually used in poultry, chickens and turkeys. And you can see that the one point that had, our highest amount of poultry had definitely our highest amount of thiamulin in it. And then sort of this intermediate sort of fit in there too. Ignore the R squared, that's clearly being driven by this one point, but it does sort of line up pretty well with what we're observing up in the watershed. Okay, I did want to end on one last point here, and that's sulfamethoxazole. Now, this is a compound that's primarily used in humans, and we found it in all of our watersheds, except for Wolf Creek, which is our residential watershed, which is really interesting. But Wolf Creek is really tiny and really doesn't have any effluent in it. And so I think that this sulfamethoxazole is really showing us that it's coming from lots of different sources, effluents from our wastewater treatment plants, as well as septic, and that it's showing up everywhere because of that. So this is important to remember that even in our watersheds that we're saying we're having a lot of animals in them or getting those inputs from that, there's still inputs from humans as well. It's coming from multiple sources. Sulfamethoxazole is oftentimes combined with trimethoprim because it's used to treat urinary tract infections. And I thank my friend Heather Pay for telling me everything she knows about pharmaceuticals because she's a pharmacist. But you can see that in this case, it wasn't a great relationship because of this one point. Maybe this is because trimethoprim was being used on animals in this watershed, because if we don't include that, we have a fairly good relationship between higher levels of sulfamethoxazole and higher levels of trimethoprim. So this lines up pretty well, indicating humans are playing a role. The last point I wanted to make was if we can link livestock and dissolved phosphorus. And I'm going to say not really. Remember, so dissolved phosphorus is the compound as the water, the nutrient that is the main cause of harmful algal blooms in Lake Erie, and its loads and concentrations in the Maumee River are really what we focus a lot of attention on. This is showing the 5-year average of these concentrations. And so you can see this familiar U-shaped curve where dissolved phosphorus increased in the 90s and coincided with the return of blooms in Lake Erie. But if we look at the cattle and hog animal numbers, we can see that this historical increase in DRP actually precedes the increase in animals by about a decade, 5 to 10 years here. So we were already up at high levels by the time we just were starting to see an increase in animals here. That makes it difficult to link livestock and dissolved phosphorus using antibiotics. But we did try to look across different watersheds, and I'm not going to focus on this a whole lot because we have a whole paper coming out on it. But basically, we see interesting patterns and dissolved phosphorus loading when it's scaled to watershed area, moving from the River Raisin all the way over to the Cuyahoga, so west to east. And that the drivers of this variation in nutrient loads ends up being land use, slope, and soil type, and animals don't seem to be playing a huge role in this. But now that we're getting some more of the antibiotic data, that's going to be some of our next steps, is trying to see if we include a little bit more information there. Maybe it can parse out a little bit more of that information for us. So our take home is that some antibiotics are detected frequently in our rivers and streams, but not all antibiotics. We had a list of veterinary compounds that were fairly common that we focused on. There are also some human compounds that we found fairly frequently. I included clarithromycin, which we didn't talk about a whole lot, but just throwing it in there so you know. These levels of antibiotics varied over time, but not very consistently. It wasn't like they were always high in July and always higher in the Sandusky River. It was pretty variable. But when we looked across the different watersheds, they do seem fairly well associated with livestock numbers, especially the particular animal that antibiotic is supposed to treat. The one human antibiotic, sulfamethoxazole, was found fairly consistently across all watersheds, suggesting that we have septic and effluent that could be contributing to the presence of that compound in our waters. And because there's not probably a great linkage already between animal numbers and dissolved phosphorus levels, I'm not sure we're going to be able to do anything more by understanding antibiotics, but we'll certainly try. And so with that, I think I'm all done. I can take any questions, or if you want to be in touch with me later, this is all my contact information.
All right, well, thank you. We've gotten some great questions during Dr. Johnson's presentation, so let me get started and ask as many as we can get to. Okay, so we got several questions dealing with just, I think, checking to see if some additional information. Here's the first question. Did you look at precipitation variability in the watersheds over the sampling period?
Yeah, so I have not looked at that quite yet. That would be a reasonable thing, of course, to look at that, or flow. That is definitely on the next step. Because this is also sort of looking at an integrated over time measurement that could certainly play a role, but it might not be as strong as if we were looking at something that was more driven by flow, like a flow weighted mean.
All right. Next question is, did you measure dissolved phosphorus in your grab samples?
So we did not do that from the specific grab samples, but we did measure dissolved, like each one of these places was a sampling location. We were collecting three samples a day for dissolved phosphorus.
All right. What are the number of humans per square kilometer, and how many of them are in the areas where the POTWs collect their waste?
I don't know that off of the top of my head. There's certainly some variation in human density through these watersheds, especially when you get to larger watersheds, you can get some larger towns and stuff, but most of these are fairly rural areas. And so there wouldn't have been, a very high density of people in any of them.
Okay.
Yeah.
Do you have any information on how long these compounds persist in the environment and remain viable as antibiotics?
Oh, that's a good question. That's hugely variable across the different compounds. Some of them can last for a long time, and some of them have half-lives that are on the order of days. And so it really depends on the compound itself as to how long it persists. I know ionophores, for instance, like monensin was one of the ones I showed, can persist for quite some time. So that's an interesting thought, though, just because, its persistence could be linked to whether we found it or not. So that's definitely something to look into a little bit more.
All right. How are the antibiotics affecting the algal blooms, or are they?
Right. Yeah. In terms of the importance of, or like how toxic or the influence of these antibiotics on any sort of aquatic organisms, that's always a hard question to answer, because studies historically, in terms of at least toxicology, have always focused on single model organisms that they're trying to measure toxicity of, and trying to understand sort of these more chronic low level exposures and how they affect any sort of natural community in an aquatic environment is difficult. So I dug into that a little bit, and I haven't been able to find a lot of very clear evidence of, that the levels that we had would have had a very strong influence, but it is an antibiotic, and the harmful algal blooms are cyanobacteria. So in a sense, you can imagine that if those levels got high enough, that they could certainly play a role in and affecting the growth of these harmful algal blooms.
All right. Let me see. Sorry, I'm looking at my questions here. One last question. We had a couple of people asking, what is the next step to this research then? Now that this is close to being complete, what would be your next steps?
Yeah, well, so for this data specifically, our next steps are to take those POSIS numbers, the integrative sampler, and turn them into, we can do some calculations to translate them into an average concentration. So that certainly is the very first thing that we want to do. And I think we're always sort of continually struggling to get the best estimate of animals that are present in our watersheds. So making sure those numbers are really good and we have the POSIS numbers translated to better make sure that that variation that we're measuring is really true. And then taking that and trying to look at some of the questions that were already asked, how important are these numbers and how do they really compare? I'm trying to find a little bit better, more recent information in terms of antibiotic levels that we see across different watersheds more recently rather than just that 2013 study.
All right. I actually have one more, sorry.
Okay.
This is going back to that persistence question. Will the regulation changes and the different practices in dispensing antibiotics to animals that you had mentioned be expected to make rapid changes to the amounts detected in the sampling, or might it take some time to see significant changes from that?
Yeah, well, since the levels are so low, I would imagine that we would be able to see. I don't think it would take like years before you would see a change. I mean, I think it would be something that there could be some buffer time, but I'm imagining that I would think that there should be a pretty detectable influence in the upcoming few years. Because a lot of changes seem like happened in 2017, I kind of think that probably what we see is already reflecting some of that, the change, some of the types of antibiotics I was expecting to see a lot of, we didn't see nearly as much of. So I think it's already having some impact, and so I think that that'll continue to happen, hopefully.
All right.
Yeah.
The only other question I had was, we had several people asking whether or not you would be willing to share your PowerPoint slides. Would that be something that would be possible?
Sure. Yeah. I can
All
do that.
right. All right. Well, I appreciate, thank you. So I'll close now. Thank you, Dr. Johnson, for your willingness to talk with us today about your research. This was really an excellent discussion. I'd also like to thank Christina Diokis for her work organizing this webinar series. I'd like to remind everyone that the survey URL for the webinar is in the chat feature, so please take a few minutes to fill that out. This webinar series is sponsored by Ohio Sea Grant and will continue in July with Bowling Green State University's Kevin McCuney, who will be talking about his research on pharmaceuticals near treatment plants in Lake Erie's Western Basin. The registration link is in the chat. Thank you again, Dr. Johnson. As always, fantastic presentation. We really appreciate it. And all the participants on this webinar, we hope that this was beneficial and hope you'll join us again in an upcoming webinar. Thanks again and have a great afternoon. Thank you, Dr. Johnson.